huge range of healthcare products is available today to treat a wide variety of medical conditions. Some medicines are made by chemical synthesis, while others, through the use of biotechnology, are extracted from living organisms. Once a patent of a chemically synthesized drug expires, competing copies, so-called generics, may be manufactured. Originator and generic products must be therapeutically equivalent, usually proven by a pharmacokinetic study called a bioequivalence study. Generics must contain the same active substance in the same amount and dosage form as the originator product, but may differ in the used excipients. Originator biologics can also be reproduced by other manufacturers after the patent expires. These are called biosimilars and should not be confused with generics due to their difference in size and complexity. Why are they called biosimilar and not bioidentical? All biologics, whether they are originators or biosimilars, are sourced from living cells, human, animal or bacterial, whose genetic material has been modified by complex biotechnology methods to produce the active substance of interest. Biologics, including biosimilars, like all human proteins, are very large and complex. Because biologics are produced in natural systems, the proteins they contain will display some variations. Although the amino acid sequence is always the same, the molecules included in the product may differ slightly from one another, for example, because different sugar molecules are attached to the molecule. This also happens to proteins made in the human body and is an example of the natural variability of proteins. This natural variability explains why biologics may vary slightly from batch to batch. An exact copy can never be produced. A biosimilar manufacturer needs to ensure their product closely matches the variability observed for the reference product. Biosimilars are very thoroughly tested, using highly sensitive methods to make sure that the product quality attributes, including the physiochemical and biological characteristics, are very similar to the originator. In the approval process, the range of variability allowed for a biosimilar is the same as that allowed between different batches of the reference product. For a biosimilar to be approved, it needs to demonstrate similarity to the originator biologic, also called reference drug, in terms of quality, biological activity, efficacy, and safety. 3 steps are involved. 1. Analytical and functional studies comparing the biosimilar and the reference drug. 2. Comparative non-clinical pharmacodynamic and sometimes toxicology studies. 3. Comparative clinical Phase 1 and Phase 3 studies in a sensitive population evaluating pharmacokinetics, pharmacodynamics, efficacy, safety, and immunogenicity. Biosimilars may differ from the reference drug in presentation, dose delivery system, and formulation. If a biosimilar has demonstrated similarity in terms of pharmacokinetics, safety, and efficacy in a clinical trial in one indication, it may be approved for use in each approved indication of the reference product, without needing separate clinical studies, provided that adequate justification is given. This is known as extrapolation of indications. Biosimilars have a comparable efficacy and safety profile as their reference drugs. But as biosimilars build on existing scientific knowledge, and the tailored regulatory pathway allows to authorize biosimilars at lower costs and risks, they can be offered at lower prices, helping the healthcare system to continue providing access to advanced therapies for all patients. Furthermore, biosimilars can also be innovative, with patient-friendlier devices or improved formulations, which allow better stability or tolerability than the originator.